Good evening. Good evening. Vanakkam, everyone. Komoda, Komoda friends. Is it raining, ne? Raining, rainy night. Komoda, hello, hello. Am I audible? Can you all hear me clearly? Hello, Mithilima, Tabhana, Jai Prakash, Santosh, Rishma. Komoda. Doing good, ne? Hi, people. Hello. Good evening. Fine, ne? Fine. Cold, rainy, ne? I'm fine. Right, people. Um, yes. Approve. Anchor. Right, people. Today we don't have to discuss the paper. It's not the paper night, neither. It's not the paper night. Theory revision night, isn't it? Theory part revision, ne? Am I correct, people? Yeah, theory part revision, no? Theory part revision. And today people will be discussing cellular respiration and we'll go with the evolution part, ne? Cellular respiration and evolution part. Cellular respiration and evolution part. Cellular. People, right, people, cellular respiration revision, cellular respiration. Let's talk about it a little bit because it's after some time, neither. After some time, cellular respiration. And then people, cell respiration is a process by which chemical energy in organic molecules such as carbohydrates are released by a stepwise oxidative process, catalyzed by enzymes, and made available in living cells in the form of ATP, right? So that's the definition. Two types of respiration present: aerobic respiration and anaerobic respiration. Neither aerobic respiration and anaerobic respiration. Right, people, we discuss aerobic respiration. We talked about glycolysis. Remember steps of glycolysis? Glycolysis, there in glycolysis, glucose, neither. Glucose, which is C6 compound, transforms into two molecules of pyruvate. Two molecules of pyruvate. Each pyruvate is 3 carbon containing compounds. 3 carbon containing compounds. There are several steps in between, but if you consider this as a single step reaction, what happens, people? 2ADP becomes 2ATP. It's actually a two-step process, right? Two-step process where 2 ATP initially spent and 4 ATP produced, but we can write it as a summary here, neither summary. And then people, during this process 2 NAD plus becomes 2 NADH. How many carbon dioxide is evolved during this process? How many carbon dioxide is going to be evolved, people, in between? Can you tell me how many CO2 is going to be evolved here? Is there any decarboxylation? Yes or no? Is there any decarboxylation process here, people? No decarboxylation, neither. No decarboxylation in between. So that's the summary of glycolysis. That is summary of glycolysis. We learn, people, the cellular respiration can be divided as aerobic. How did you divide cellular respiration? How do you divide cellular respiration? How do you divide cellular respiration? Cellular respiration. How do you divide cellular respiration? Cellular respiration, we can show some steps. Cellular respiration, we can show some steps. Right? Some of the steps of cellular respiration. we can take that cell respiration, some steps happen in the cytoplasm. So where glucose is transformed into pyruvate, pyruvate, and we call this as glycolysis, right, people? This step we call as glycolysis. That is glycolysis. And then pyruvate has two fate, right, people? Pyruvate has two fate. Pyruvate has two fate. One is pyruvate can undergo the aerobic respiration. This is glycolysis. Glycolysis. This is glycolysis. Pyruvate can enter into aerobic respiration, where pyruvate enters into the mitochondria. This is mitochondrion, and pyruvate enters into mitochondrion and then undergo series of reaction. What do you call that series of reaction, people? You call it as aerobic respiration. No, I'm wrong. Series of reactions occur here inside the mitochondrion, right? And glycolysis and these events which happen inside the mitochondrion, we can take it as aerobic respiration. Glycolysis and all these events we take as aerobic respiration. Aerobic respiration, right? So we learn, people, glycolysis has a common step, right? Glycolysis has a common step. So if you take here the Santosh's link reaction and the Krebs cycle occur inside here, yeah. This is Krebs cycle, link reaction, the link reaction occur in between. And then, people, something happen on the mitochondrial membrane, what do you call that? Oxidative phosphorylation, right? Oxidative phosphorylation. Oxidative phosphorylation occur there. And so this is glycolysis together with these steps. We take as, people, the aerobic respiration, right? And then, people, there is another fate. What is that, people? Glycolysis in absence of, now this event we said, people, happen in presence of molecule oxygen, neither. This event happen, people, in presence of molecule oxygen. This happens in presence of molecule oxygen. So when molecule oxygen present, molecule oxygen O2, molecule oxygen present, this happens, right? And then, people, pyruvate transforms into lactic acid, right? In the lactic acid fermentation, sometimes, people, it can transform into acetaldehyde, and then people, acetaldehyde, and then this acetaldehyde become ethanol, acetaldehyde become ethanol, and that happens in the ethanol fermentation, lactic acid fermentation, ethanol fermentation together. Here we call as, people, fermentation. This is fermentation, right? So if you take, people, this whole process, if you take this whole process, glycolysis and fermentation together, we can say aerobic respiration, right, people? Aerobic respiration. So you understand now, people, aerobic respiration, anaerobic respiration, anaerobic respiration, right? This part is called as the anaerobic respiration. So glycolysis, you understand, people, is a common step. Glycolysis is a common step for both aerobic respiration and anaerobic respiration, right? Glycolysis is a common step for aerobic respiration and anaerobic respiration. We say people this fermentation process or this anaerobic respiration happen when O2 absent neither. When O2 absent, the this event happen when oxygen is not present. And then people sometimes there can be nitrate neither. Sometimes people nitrate NO3 minus. NO3 minus can be there, nitrate. And even if nitrate present, people we say nitrate respiration because not molecular oxygen, but nitrate can participate. We take it as anaerobic because you have to understand molecular oxygen, right? Presence of molecular oxygen only we consider as aerobic respiration. If any other form of oxygen is available, nitrate, still we take it as people anaerobic respiration. But you have to understand there the oxidative phosphorylation, Krebs cycle, and the link reaction occur. Right? When nitrate involved, still link reaction occur. Krebs cycle occur. Oxidative phosphorylation occur. Only thing is molecular oxygen is not there. Right? So what do you call that? Aerobic or anaerobic? Still anaerobic respiration. But thing is, people, it's little out of our syllabus. Little out of our syllabus. We talk about the nitrate respiration later in microbiology lesson, but we don't go into these details. Right? We don't go into these details, but therefore you have to understand it's very important to say molecular oxygen, and you have to say molecular oxygen because molecular oxygen has a weight. Neither molecular oxygen, saying molecular oxygen has a weight. All right, people. Okay, so this is what we discuss, and this is what we discuss, and this is what we discuss. Right, aerobic respiration, process of synthesizing ATP from respiratory substrates such as glucose. The process of synthesizing respiratory substrates such as glucose in presence of molecular oxygen is known as aerobic respiration. So molecular oxygen is known as aerobic respiration. Glucose is found in the as the major respiratory substrate. Glucose is available as a major respiratory substrate. So here, people, this process consists of three main steps. The aerobic respiration. What are those three main steps? Glycolysis, pyruvate oxidation, and Krebs cycle. And then oxidative phosphorylation, or else what you call the electron transport chain. So we start with the glycolysis. People take place in the cytosol of the cell because all enzymes that catalyze the reaction of these glycolysis are formed in the cytosol of the cell. So where are the enzymes available? In the cytosol. This process does not depending on molecular oxygen. No need of oxygen for that. So oxygen is actually needed, people, for the last step. Oxygen is needed for the last step. Right? Last step for the electron transport chain is the requirement of molecular oxygen is there, people, for the electron transport chain. Right? And then people, 2 ATP molecules are used up initially, and then 4 hydrogen atoms, 4 hydrogen ions and electrons released from molecule of glucose breakdown and reduced to NADH and produce 2 NADH molecules. That's important, neither. 4 hydrogen ions and electrons are released. 4 hydrogen ions and electrons are released, which involving in forming 2 NADH, produce 2 NADH, right? That's like a special point. You to understand, people, this one is having only 1 hydrogen. So 2 of them is having 2 hydrogen. So what happens to the rest of the 2 hydrogen, people, is a question. What happens to the rest of the 2 hydrogen is a question. You have to understand, people, it's present anyway within this molecule. NADH, when we say, people, NADH is holding 2 more hydrogen by ionic bonds. They are holding 2 more hydrogen by ionic bonds. In our syllabus, this holds 2 more hydrogen. Each NADH molecule is holding two hydrogens by ionic bonds. We don't discuss, people. We don't discuss in that our syllabus. We don't go into that depth. But what you have to understand, every NADH molecule, right? Visibly, there is one hydrogen present in NADH. There, one hydrogen is there, but you have to understand it is capable of holding another hydrogen. So each NADH molecule has two hydrogen in it. Two hydrogen in it. So both NADH molecules have four hydrogens in it. Got it right? Right. So yeah, that's little extra information, people. Little extra information, right? And then people, since two ATP molecules were used up for the initiative step, the net yield is two ATP molecules. Only when oxygen is present, pyruvate molecule will enter into the mitochondria, and further steps will take place. Oxidation of pyruvate or the link reaction. These two pyruvate molecules enter into mitochondrion by active transport through the membrane. Active transport through the membrane. Very important part. Active transport through the membrane. So how many ATP required for these people? 2 ATP needed for this transportation process. Right? So as pyruvate enters into the mitochondrion, as these pyruvates enter into the mitochondrion, they are now penetrating the mitochondrial membrane. They are now passing through the mitochondrial membrane. As they pass through the mitochondrial membrane, this process is an active process. So here two pyruvate comes to the mitochondrial matrix, and this process requires people 2 ATP. It requires 2 ATP. But we don't write it here. We don't write this here. Reason is that 2 ATP, right, expenditure is not equal for all. Right? We take that 2 ATP, people, except for heart and liver cells, except heart, muscles, and the liver cells. Rest of the ones we have to deduct this 2 ATP spent on pyruvate transportation. Right, people? Now what happens? This 2 pyruvate transform into 2 acetyl. And then the people, this step is a decarboxylation, so that 2 carbon dioxide is going to be released. Upon conversion, you have to understand acetyl is having 2 carbon. And then you learn, people, during this process, something come and join here. What do you call that? Coenzyme A. Coenzyme A come and join. And then it forms, people, acetyl coenzyme A. Acetyl coenzyme A, right? So we talk about acetyl coenzyme A, right? Come and join here. Acetyl coenzyme A. Right? Acetyl coenzyme A. So let's take this process once again. This is mitochondrial membrane. This is mitochondrial membrane. And then here, people, pyruvate. Here, pyruvate. 3 carbon containing compound enter into the mitochondrial matrix. Pyruvate. 3 carbon containing pyruvate. Then transform into 2 acetyl, 2 pyruvate. We'll take it as 2. 2 acetyl coenzyme A. 2 acetyl coenzyme A. During this conversion, 2 carbon dioxide is going to be eliminated. During this conversion, 2 NADH is going to form. During this conversion, 2 NADH is going to form. Right? 2 NADH is going to form. Right? And then, people, this acetyl enters into a cyclic event. This enters into a cyclic process. I can't remember. What should I write here, people? Can you help me a little bit? What should I write here? Here. What should I write here? This event, cyclic event. What should I write here? Someone says OAA. And what is the product result from here? It is people citrate. Citrate or citric acid? What did you write? Citric acid. Okay, citric acid. How many carbon in citric acid? 6 carbon in citric acid. it transforms into 5 carbon compound, C5 compound. C5 compound becomes C4 compound, 4 carbon containing compound, another 4 carbon containing compound, another 4 carbon containing compound. So series of 4 carbon containing compounds are formed here, and citrate to this one, people, it forms 2NADH. During this conversion, 2NADH. And then here also, people, we say that 2NADH. And then in the next step, we said 2ATP, 2ATP. The next step, we said, people, 2FADH2, 2FADH2. And then it forms another C4 compound, right? And then there is a small gap here where, according to the resource book, no specific event there. And then people, 2NADH is going to form, 2NADH is going to form, right? So a couple of steps are there, right? So what do you call this process is Krebs cycle. So where the decarboxylation take place? Here decarboxylation take place, 2 carbon dioxide if the cycle run 2 times. And here, people, decarboxylation occur when the cycle run 2 times. So as the cycle run, Krebs cycle, as a cycle run 2 times, we can see that, people, 2 carbon dioxide here, 2 carbon dioxide there, 2 times carbon dioxide is going to be eliminated, right? And plus, people, those 2 carbon here, those 2 carbon altogether, 6 carbon dioxide. So that the carbon present in the entire glucose molecule is now eliminated during this stepwise event. And then, people, what happened next is the interesting event. What is that? Electron transport chain. Next event is electron transport chain. So let's see, people, what happens in oxidative phosphorylation, or else what you call this electron transport chain. Electron transport chain. So we learn, people, electron transport chain is a cascade of reaction, one after the another, one after the other, series of reactions occur. And then the important part is, people, there are a couple of proteins present. There are a couple of proteins present, electron transport proteins. This NADH is fed here. Let's take that this NADH is fed like that, becoming NAD plus. It becomes NAD plus. This NADH becomes NAD plus, eliminating hydrogen, limiting hydrogen and electrons. And these electrons are passing through series of electron carriers, right? Lowering its energy. They pass through series of electron carriers like this. And finally, people, reaches to an enzyme here. And then this process is pumping H plus ions. This process pump H plus ions into the space, right? Like this, into the lumen, not the lumen, into the intermembrane space. H plus ions are transported like that. We don't discuss this. What you know is that some H plus transportation occur. And as these H plus ions are returning back, each H plus ion is capable of producing ATP. Right. So in general, people, simply we say that as one NADH molecule break down into NAD plus, and as it release a pair of electrons, when these electron pairs pass through a series of electron transport proteins, 2.5 ATP is produced. 2.5 ATP is produced. Forget about the rest of the part. 2.5 ATP is produced for every NADH. So if you calculate the total number of NADH, 2 NADH, 2 NADH, 2 NADH, 2 NADH, how many NADH available, people? How many total NADH available? 10 NADH, 8 NADH present, but together with the glycolysis, NADH, it's 10. And then therefore, how many ATP produced? 2.5 each, 10, it is 25. And then, people, FADH2, how many? 2. As FADH2 enter this process, Every FADH produce 1.5 ATP. 2 FADH therefore produce 3 ATP. 25 plus 3 is 28 ATP. So the oxidative phosphorylation only, alone oxidative phosphorylation is forming 28 ATP. 28 ATP plus 2 ATP produce here 30. Plus people glycolysis 2 ATP, glycolysis 2 ATP 32. 32 ATP is produced for every glucose molecule. But you know, this number is actually 30. Right? This number is actually 30 for all other organisms except heart and the liver cells. Heart and liver cells. Heart and liver cells, people, the number of ATP produced in the oxidative phosphorylation is little higher. So therefore, these two ATP transport from the pyruvate transportation can be neglected. Is that clear? They also need ATP for this pyruvate transportation, but they produce little more ATP in the oxidative phosphorylation. Therefore, we ignore the amount of ATP spent on here because the ATP production is not 28, 30. So we say that that 2-NADH, right, can be neglected. So we don't say that. We don't try to make the details that complicated, but without that we can describe it. Right? We describes the process. Haridalamai, right? I think this is all right. This is okay. This is okay. All right, people. Then we come into the. Then we come into the. Then we come into the. Then we come into. Then we come into the citric acid cycle. There something important is there about the pyruvate. This process is important. Pyruvate, people, 2,3,2C3H4O3. This year remember, right? Pyruvate. This one. It become 2 coenzyme A plus NAD plus citric acid cycle. This take place in the matrix of mitochondria using specific enzymes. As the first product of this cyclic pathway is citric acid, known as the citric acid cycle. The pathway was discovered by German-British scientist Hans Krebs. Citric acid contains 3 carboxylic acid groups. This is called as tricarboxylic acid cycle also. And then citric acid undergoes series of enzyme catalyzed reactions to generate oxaloacetate by releasing 2 carbon dioxide molecules by decarboxylation reaction. So what they say, decarboxylation reaction, and 1 FADH2 and 3 NADH will be generated as a result of oxidation reaction. There are products of single acetyl group led into citric acid cycle, and hence these numbers should double when the yield for glucose molecule is considered. So we learn this, we learn this. That's simple, people. Electron transport chain, fine. And this is the summary, fine. Anaerobic respiration. Now let's talk about anaerobic respiration. Anaerobic respiration. So there are 2 types of anaerobic respiration. What are those, people? Ethyl alcohol fermentation and lactic acid fermentation. Ethyl alcohol fermentation and lactic acid fermentation. Ethyl alcohol fermentation, like in aerobic respiration, the first step is glycolysis. And then this pyruvate involves in 2 steps. First step, pyruvate is converted to acetaldehyde, releasing carbon dioxide. Second step, acetaldehyde is reduced using NADH to produce in glycolysis. Therefore, final hydrogen acceptor is ethyl alcohol. That's important. That is important. We have to talk about the final hydrogen acceptor. In the final hydrogen acceptor. Now here people in ethyl alcohol fermentation, acetaldehyde, final hydrogen and the electron acceptor is this one. Final hydrogen and the electron acceptor is acetaldehyde. And then in lactic acid fermentation, I'll take another new chart. So here people, this is pyruvate. This is glucose. It becomes pyruvate. Neither. Pyruvate. Pyruvate, 3 carbon pyruvate. When 3 carbon pyruvate transform into lactic acid during this conversion, you know, glucose to pyruvate 2NAD plus becomes 2NADH. Neither. So this is going to be reversed. This is going to be reversed. That's the whole purpose of this fermentation, to make NAD plus. Process is reversed. Right. Therefore, what is the final hydrogen in the electron acceptor? Pyruvate. Pyruvate captures these H plus ions and electrons from NADH. But people, when you take the ethyl alcohol fermentation, first it produces acetaldehyde. Acetaldehyde. That acetaldehyde transforms into ethanol. Ethanol. Now you know ethanol is C2 compound. Acetaldehyde also C2 compound. How about lactic acid? This is a C3 compound. C3 compound, people. Right? So during this process, acetaldehyde formation, decarboxylation occurs. Lactic acid formation, there is no decarboxylation. Lactic acid formation, there is no decarboxylation. Here, no decarboxylation upon forming lactic acid. Why? Pyruvate has 3 carbon, lactic acid has 3 carbon too. So no decarboxylation. But here in ethyl alcohol fermentation, ethanol has 2 carbon, whereas pyruvate has 3 carbon. So there is a decarboxylation step. Carbon dioxide is going to be lost. And then we will acetaldehyde is a 2 carbon product, it will become ethanol. So during this, the same conversion happened. Same conversion happened. What happened there, people? As acetaldehyde become ethanol, 2NADH become NAD plus. And here, the final hydrogen acceptor is acetaldehyde. This hydrogen available in NADH is captured by acetaldehyde. So we say final hydrogen and the electron acceptor in ethanol fermentation is acetaldehyde. Hope this is clear for you. Hope that part is clear. We have discussed these things before, right? But just for your information, for the second time, second time, 10th time, we have discussed these things multiple times. So fine, it's fine. All right. So this is it. So let's talk about the lactic acid fermentation as ethyl alcohol fermentation. As in ethyl alcohol fermentation, glycolysis takes place in the first step of lactic acid fermentation. Therefore, 1 molecule of glucose is produced from 2 molecules of pyruvate, 2 molecules of ATP, and 2 molecules of NADH. The pyruvate is reduced directly by NADH for lactic acid as the end product with no release of carbon dioxide. Pyruvate is reduced directly by NADH for lactic acid and no release of carbon dioxide. Therefore, final hydrogen acceptor is pyruvate. Certain fungi and bacteria carry out lactic acid fermentation. Okay, some bacteria and fungi. Sir, if both have the same process, why we need 2 types of fermentation? What is that? So if both have, Hemanta ask, sir, if both have same process, why we need 2 types of fermentation? That's like different types of metabolism in different organisms, isn't it? Right? Different type of metabolism in different organisms. Some are producing lactic acid because they have the enzymes and they have the ability to now lactic acid once it is produced people need lactic acid. Now for an example, suppose you produce ethanol. Suppose Hemant, let's take that you produce ethanol instead of lactic acid fermentation, right? So when you carry out anaerobic respiration, you are frightened to a to an animal and then you run away from the danger. Instead of making lactic acid, you produce ethanol, right? Then what will happen when you produce more and more ethanol by anaerobic respiration as you try to get away from danger, right? As you carry out anaerobic respiration, if ethanol level goes higher, what will happen to you? You get doped, Neil. You get doped, right? And you won't be able to run away from danger. So therefore, what is produced by different organisms is depending on its capability and its metabolic uh, design, isn't it? Right? If we have ethanol instead of lactic acid, we won't be able to function effectively because we have nervous system and nervous system get affected by ethanol, right? But lactic acid don't do that, right? Lactic acid is basically produced inside the muscles and then they are transported through blood and then people metabolize easily by the liver, right? So it's the different abilities. We can't expect that every organism on earth to have the ethanol fermentation, isn't it? Understood, right? Okay. Now, people, we learn about the respiratory quotient. Respiratory quotient is this proportion needle. The ratio of carbon dioxide evolved. So, RQV is volume of carbon dioxide to volume of oxygen. Respiratory quotient, right? So, we learn people the use of other compounds in respiration. Proteins can involve in proteins first transform into amino acid. Amino acid undergo deamination like this. Amino acids undergo deamination. It become deamination. So ammonia is going to be evolved. Remaining carbon skeleton enters in different steps. Remaining carbon skeleton can enter at different steps. Right? Remaining carbon can enter at different steps. Sometimes people, they can become acetyl coenzyme A. Sometimes they can enter into the Krebs cycle. Sometimes they transform into pyruvate. Then people, if you take the involvement of fat in aerobic respiration, glycerol can become people G3P, fatty acid can become acetyl coenzyme A, right? Fatty acids can become acetyl coenzyme A, right? Other than the what you call glucose, other carbohydrate can take this pathway. Other carbohydrates, other monosaccharides, other polysaccharides, they can become sugars, and sugars can involving in forming glyceraldehyde. So the different types of carbohydrates can be metabolized. Different types of carbohydrates can be metabolized, not only glucose, right? Not only glucose. So this is a small summary of the cellular respiration process. Summary of cellular respiration process. Time to time, people, we have to go through different theory paths, right? Different theory paths. When you forget, my job is to right to help you to revise those sections, right? I'm showing you certain parts in the resource and helping you to go through them. People, my second target for the day is the condition of the early earth before life. We discussed this before, I know that. Conditions of early earth before life. So we'll go through that part a little bit, people. Let's go through that part a little bit. Let's go through that part a little bit. Yeah, I think, no. Right. I think this part is better. See if they can see it clearly. We talked about people, different eons we discuss. Before we talk about eons, we talked about check this one, the early Earth and our planet and the solar system formed around 4.6 billion years ago. The first atmosphere was probably thick with water vapor along with various compounds released by volcanic eruptions, including nitrogen and its oxides, carbon dioxide, methane, ammonia. You have to remember these, huh? What were the early gases, people? Nitrogen, including nitrogen and its oxides, carbon dioxide, methane, ammonia, hydrogen, and little oxygen. Little oxygen were there in the early atmosphere. The atmosphere was turned to be a reducing one later. It was cooled down and water vapor condensed into the oceans. Much of the hydrogen escaped to the space. Volcanic eruption, lightning, and extreme UV radiation, hydrothermal vents, and alkaline vents. And along with the Earth reducing atmosphere favored the synthesis of organic molecules. So the first organic molecules were formed, and then we discuss, people, the steps neither. Evolution of biological diversity, biochemical theory of evolution. There we said, people, we have evidence to say that the formation of the first cell, protocell. So biochemical theory of evolution. Direct evidence for the life on early Earth comes from fossils of microorganisms about 3.5 billion years ago. Observations and experiments in chemistry, geology, and physics have provided evidence for the appearance of first living cell. The theory of biochemical evolution arose from the hypothesis based on the chemical and physical process on early Earth. The emerging force of natural selection could have produced a first cell through sequence of 4 main stages. All right. Look at this sequence of 4 main stages. First one, the atmospheric condition of early Earth facilitate the abiotic synthesis. So abiotic synthesis, small organic molecules were formed by people. Inorganic compounds in the early atmosphere had resulted, people, organic molecules, right? And what we say, people, the next step is these organic compounds were polymerized to form small organic molecules. And from them, the organic macromolecules had formed. So amino acid has transformed into proteins. Nitrogen bases, sugars, and phosphates have transformed into nucleic acids. And then organic macromolecules were packed into membranes to produce the protocell, the third event. The formation of the protocell is the third event. And finally, we say nucleic acid gain the self-replication ability. Right? Nucleic acids obtain the self-replication ability. So first step, what we said, people? First step is facilitating abiotic synthesis of the simple organic molecules from inorganic molecules, polymerizing of the simple organic molecules to form macromolecules, macromolecules packed into membranes to produce protocells, and finally, nucleic acids obtain the self-replication ability, right? And then given the cell, right? Hemant, later Earth was cooled down and the water vapor condensed into ocean. Why much of the hydrogen escaped into the space? Now, thing is, somehow we have to say the hydrogen cannot continuously present because it's a fuel, and it's actually it's flammable. So we can't say that the atmosphere is having too much of hydrogen when volcanic eruptions occur. We can't say that the atmosphere is having a lot of hydrogen. Hydrogen is like a bomb. So in every eruption or with every lightning, people. Otherwise, it's going to be like fireworks throughout the whole Earth atmosphere. So hydrogen level has gone down. So we believe at one point there was so much of hydrogen at the beginning of the formation, right? But then gradually this hydrogen level went down, and then there was a little oxygen, but anyway, there were no too much of hydrogen inside, but it was a reducing atmosphere, right? And then gradually the release of oxygen by the photosynthetic organisms that gradually transform the reducing atmosphere into an oxidizing atmosphere. Is that clear? Right? I think it doesn't set, right? It doesn't set to say that the hydrogen were continuously present in high level in the atmosphere. Does not set, because they try to build up a story. They try to build up the story. Sometimes people, this formation of organic molecules from inorganic compounds, formation of inorganic compounds were transformed into simple organic compounds, right? This process does not happen in less hydrogen. Maybe when more hydrogen present, that process happened effectively. But later on, people, when they describe the formation of further macromolecules and all, people having too much hydrogen may be not set. Neither. There can be many reasons for them to consider like that, considering the chemistry behind, right, of these reactions. Right? When someone says, oh, no, it can't happen in high hydrogen level, then they say that maybe then hydrogen escaped. There is always possibility. So they try to make up the story, because no one knows for sure. No one knows for sure. We only know the outcome of it. So we try to describe the situation, people. To match the situation, we build up the story. That is how it goes, right? So that is how it goes. Maybe they have some more evidence, maybe some of the other evidence, geology and other evidence, maybe they have people to say that there were more hydrogen earlier and there were less hydrogen later. So they use that to the story. Right? They make it to the story. You can make a story like that too. When you go to school, then someone was there, and then you see a cigarette butt or something. So you can make a story, right? Setting that to that your innocent friend, without you try to set the evidence for the story. That's all. Okay, so people, origin of protocells. So we'll be discussing now, people, the origin of the protocell. Haldane suggested that the early oceans were solution of organic molecules and called the primitive soup. So there was a primitive soup in the early days. And recent studies related to the volcanic atmosphere and the alkaline vents show abiotic synthesis of organic molecules. So recent studies are relating the organic molecules had formed in more reducing volcanic atmosphere, or maybe they were formed in people alkaline vents. Another source of organic molecules may have been brought by the meteors. RNA accumulated into lipid-bound vesicles and formed protocells, which exhibited enzyme catalyzed activities and were able to grow, replicate, and evolve. The early genes and the enzymes would have been RNA. These RNA molecules enable the self-replication of RNA. Other molecules that were in the primitive soup were also collected in the protocell. Growth occurs by addition of lipids into the membrane by collision of vesicles. When the protocell become too large, it divided to form two protocells with RNA included. Now, people here they say, right, the protocell formed by gathering of these materials in the early soup, and then these protocells grew bigger and then break into pieces. But still, people, the protocells were not capable of transferring characters from one generation to the next. That is also needed for the life, isn't it? The features has to go from one generation to next. And that goes, people, with the incorporation of the RNA. So the early enzymes, and also the genetic material would have been RNA. That is what they say here. Right? So then the space is a vacuum, right? Where is that escaped hydrogen? Where is that escaped hydrogen? No, in stars also this fusion reaction take place. So we can't say that the space is entirely a vacuum. How about then the sun? Yeah. How about then a sun? There is hydrogen in the sun. So maybe that gravitational pull hold these gases to a certain the celestial body. That can happen, right? It can form atmosphere somewhere else, right? If that gas escaped to another celestial body. So good questions. So you ask right tricky questions today, right? So yeah, that's good. I think you have now time to think about all these other things. Possibilities are there. How about the moon and other things? They can have light atmosphere too, right? But I don't say, right? It says that it escaped, right? Because you can't say that it's like over there. Or maybe burn out neither. Maybe burned out. Right? But it doesn't say, it says escape, right? All right. Then origin of the photosynthetic organisms. Origin of the photosynthetic organisms. But if you are interested, you can read that Campbell's biology. Campbell's biology, that they give good description. So how does the sun gain so much of energy? That's a nuclear fusion reaction. Nuclear fusion reaction where the hydrogen becomes helium. So that nuclear fusion, not fission, nuclear fusion reaction. At the point of the formation of the earth, there were only two elements, hydrogen and helium. So that the stars, the energy is this nuclear fusion reaction is releasing energy. Right? So that was the fuel. That is the fuel to run the universe. Yeah, and it's the only fusion reaction. Even in our atomic energy plants or in bombs, also we don't use that fusion reaction. Right? Normally we use a fission, right? Fission. All right. Okay. So origin of photosynthetic organisms, fossils of the first atmosphere. Shall we go to this one, people? This one, this one we talk about. You can see the board, and you can see the board. I think this is good enough. We can divide the whole time period into several eons: Hadean eon, Archean eon, Proterozoic eon, and Phanerozoic eon. Phanerozoic eon, right? So let's start with the first one, people. Hadean eon, origin of Earth 4.6 billion years ago. Archean eon, oldest known rocks on Earth 3.5 billion years ago. Oldest fossils of the prokaryotes had appeared. Neither. So this is the first evidence that we have for the life. 3.5 billion years ago, the formation of prokaryotes. 2.7 billion years ago, in the same Archean eon, concentration of the atmospheric oxygen began to increase with the formation of the photosynthetic prokaryotes, right, people? So should we memorize about the facts, eons and eras? Resource book says that you don't have to remember them, but I'm telling you to memorize them, right? Because you're learning so many things for A-level biology. This is not, people, much for you, this you can tolerate easily. They have asked questions in the MCQ paper and all. Structured essay paper also, they have asked questions. They don't want you to study it, right? So you decide, yeah. So there were questions in your paper number 10 also, yeah. And there were questions in Past papers also you can check 2019 LL paper. Couple of papers are there. They have asked questions. Right? You better study. Then people Proterozoic eon 1.8 billion years ago. Oldest fossils of eukaryotes appear 1.2 billion years ago. Diverse algae had formed. Right? Diverse algae had formed. So up to which content we should study? We should study for upcoming exam. Unit 2 and Unit 3 only. Unit 2 and Unit 3 only. Only 2 units. I don't expect more from you, more than that. Unit 2 full. Unit 3 full. That's it. Right? Okay. That's easy, ne? Right? So here, okay, so diverse algae, the algae formed people 700 million years ago. Diversification of algae, ne? Diversification of algae 700 million years ago. No, 1.2 billion years, ne? All else fossils of eukaryotes 1.8 billion years, 1.2 billion years. Diversification of algae 700 million years ago. Soft-bodied invertebrates had appeared 670, what we said, people, 670. The ancestors of arthropods, chordates and other animals formed 670. Right. And then people, we come to the Phanerozoic. You don't have to remember the periods now. Cambrian, sudden increasing in diversity of many animal phyla. And then in Ordovician, marine algae became abundant. Colonization of land by diverse fungi, plants and animals. Plants and animals, they colonized the land 470 million years ago. 470 million years ago. Right? 470 million years ago. 425 million years ago, origin and diversification of vascular plants. People, this origin and diversification of vascular plants 425 million years ago, this was actually mentioned in the plant diversity part. There was a question regarding that. 425 million years ago, origin of vascular plants, that I have taken people from the resource book. I have taken from the resource book that diagram. This one, this page diagram. 365 million years ago, diversification of bony fish, first tetrapods and insects appeared. 305, that also I have taken people from the resource book. The amphibians dominated. Extensive forest of vascular plants had evolved. First seed plant had appeared. Origin and radiation of reptiles. So I drew a cartoon for that. Reptile eating the seed. And then people comes the next period, Permian. Origin of modern group of insects. Extinction of many marine and terrestrial organisms. Permian, people, there was a mass extinction. Diversification of vascular plants. And then people come to the Mesozoic era. Triassic cone-bearing plants. Dinosaurs evolved. Origin of mammals. Hey, look at this cartoon. Right? Look at this cartoon. Right? This cartoon. Cone-bearing plants, ice cone, dinosaur, and the mammal. And then people came, the Jurassic. Gymnosperms continue to be the dominant plants. Dinosaurs dominant. Birds evolved, that is not actually mentioned in the resource book. And then people, Cretaceous, end of the Mesozoic. right? End of the Mesozoic, flowering plants, angiosperms appeared and diversified. Many organisms, including dinosaurs, became extinct. And then people, the Cenozoic, the Cenozoic period. Cenozoic period. So Cenozoic, what you say, people? Cenozoic, major radiation of mammals, birds, and pollinating insects. Domination of angiosperms increased, and their radiation continued. Radiation क्या मतलब है? Rapid evolution, ne? And dominance of angiosperm increased. Origin of many primate groups. They continue radiation of mammals and angiosperm. Earliest direct human ancestors were formed. And then people appearance of bipedal human. And then finally they say origin of the genus Homo. Origin of the genus Homo. Yeah, so those were the events, right? Those were the events that had happened over the past time period, right? So I have questioned these things, people. I have questioned these things, some of those parts in the tute. So we talk about these theories of evolution too, ne? Theories of evolution. Two theories of evolution we discuss, people, the Darwinism and the Lamarckism. Neither Darwinism and the Lamarckisms were the two theories, right? Theories of evolution. Changing of genetic composition of population from generation to generation over a long time period is resulting people evolution. Lamarck's theory, what he said, two ideas, ne? Use and disuse. Use improves, disuse deteriorate, ne? And these characters, the build up characters inherit. That is Lamarckism, ne? Use improve, disuse deteriorate. And these characters inherit. So when will be the exam held? We said April 11th, neither. One of you said, sir, please postpone it further for April 18th. April 18th, we don't have a class. Ne? April 11th, another 7 days later, our exam is over. Mithulima says exam is over. 11th was the planned date. Why 18th? Someone is requesting to postpone it. I think 18th is still that New Year vacation, ne? New Year vacation, ne? New Year vacation, right? New Year vacation goes to 18th, I think. 18 is there is an auspicious time for Aratine Rekrakshasana Pitatwima Kilakatine. Neither. Etakam bus karane. No buses. Right? No buses on 18th. Bus yanne after 18th, neither. Isn't good to keep exams on 11th. I think it's better, ne? 11th is better, ne? Neither. 11th. Sorry. 11th, I think better. 11th better. So, two principles here, ne? Use and disuse and inheritance of acquired characters. Darwin's theory, people, there are several ideas, ne? Overproduction is there. And then population varies in characters. See here, overproduction, ne? So many children. So many people, ne? Different abilities. Samarakiyum gasnagina, samarakiyum padankarna, mukut karanetino tinna, ne? Some go to the gym and all, ne? So population vary in characters, ne? Certain character is better for survival, ne? Rather than studying and then climbing and not doing anything, people neither. Bodybuilding has more demand now, ne? Right? Bodybuilding has more demand, ne? It was say more springs. The more offsprings are, what is that? More offsprings, certain characters are better for survival. And then what happened, people? Survival of the fittest. And then the natural selection, ne? Fourth event is what we will natural selection. Then as a result of the natural selection, finally we can see that there can be more members. More members may enhance the abundance of favorable characters at the end. May enhance the abundance of favorable characters at the end. That's actually the Darwin's idea. Darwinism. Now we say neo-Darwinism. What is neo-Darwinism, people? Charles Darwin's theory of the natural selection, Mendelian genetics, for the understanding of the population genetics. What it says, neo-Darwinism? Neo-Darwinism says it denotes the integration of the Charles Darwin's theory of natural selection, Mendelian genetics as a basis for the biological inheritance and the knowledge of population genetics, right? So that's it. And we discuss some the past theory past. Okay, then people, time to wind up. And then thank you very much for your participation in the night discussion, right? And I'll be meeting you again, people, on Saturday, right? Saturday at the same time, right? 7:30 a.m. Come to the class 7:30, people. Some people are now getting late. I'm coming sharp at 7:30. We are starting the class at 7:30. And this week, class people, we are going to learn something very interesting. What is that, people? Secondary growth. And we're going to do some questions on secondary growth. So don't miss the class, right? And then people, make sure that you answer paper number 10. You have to submit the paper also, right? So thank you very much. Big salute for all of you, right, who join tonight. And then bye-bye.